At Selling Biosciences, we are collaborating with physicians and scientists to develop the most sophisticated technologies and techniques for maximizing the collection, concentration, and delivery of autologous blood and bone marrow aspirate. Collection. The hand device is being developed to assist physicians in mimicking the bone marrow aspiration techniques of Dr. Philippe Hernigot, who reports increases of up to 300% in CFUF, or progenitor cell concentration in BMA, by collecting small aspiration volumes in 10 cc syringes. According to Dr. Hernigot, the highest concentration of progenitor cells in bone marrow is in the iliac crest. The aspiration needle is advanced into the trabecular bone of the crest. With a rapid pull of the 10 cc syringe, 4 to 6 cc's of rich BMA is collected. Compared to a 60 cc pull, this series of shorter syringe draws results in a higher quality collection. The physician will rotate or pull the needle out 1 to 2 centimeters between aspirations to sample from different areas adjacent to the single channel. This technique allows the physician to collect BMA from many areas of the marrow without removing the needle or exchanging syringes. Concentration. The ART BMC bone marrow concentration system provides separation and selection of different cellular portions of centrifuged bone marrow aspirate followed by ultrafiltration of plasma. After a 15-minute centrifuge, there are 3 distinct fractions: the heavy red blood cells, the buffy coat containing white blood cells and platelets, and platelet-poor plasma. The buffy coat at the RBC plasma interface represents less than 1% of BMA volume and includes multinucleated cells, mononuclear cells including progenitor cells, and platelets. Unlike other systems, the ART BMC allows the user to segregate the 3 phases independent of the patient's red blood cell volume. In this sense, the ART BMC is a miniaturized laboratory in a single-use, sterile device. First, the collection window is lowered above the buffy coat and the platelet-poor plasma is extracted from the main chamber to a syringe at the B port. The plasma may be sterilely stored in the reservoir. Next, the collection window is lowered just below the buffy coat to ensure that the entire buffy coat is extracted in a 3 to 5 cc volume. If additional BMC volume is desired, the user may access the platelet-poor plasma to extend the volume without a sterile break or exchanging syringes. Selling Biosciences offers devices with a proprietary and FDA-registered integrated filter system for the ultrafiltration of platelet-poor plasma. By pushing the plasma from one syringe through the hundreds of hollow, nanoporous fibers, water and salt ions are pushed out while proteins and cells are further concentrated. The fluid may be concentrated by passing through the filter until the desired volume is obtained, as low as 3 cc's. This ultrafiltration enriches the concentration of most proteins. The delivery of autologous therapies is the practice of medicine determined by the physician. Physicians often inject the concentrated fluids, like PRP, through needles as large as 11 gauge and as small as 25 gauge. In other instances, surgeons have reported the application of autologous fluids onto the wound or tissue. Other surgeons choose to combine autologous fluids with graft materials for implantation. Solum 4 is a novel bone grafting material. FDA cleared as a bone void filler when combined with autologous bone marrow aspirate. Each granule is composed of fused nanohydroxyapatite particles with a porous superstructure resulting in high surface area per volume and minimizing the mass of graft per volume desired by the physician. When combined with bone marrow, the gelatin carrier quickly gels to provide desirable handling characteristics and creating a moldable substrate for packing bony voids and defects. Selling Biosciences collaborates with leading physicians and scientists in developing sophisticated products and techniques of the highest quality for maximizing the recovery and concentration of autologous cells and proteins from bone marrow aspirate and peripheral blood while maintaining high safety and sterility. Call or email today to schedule a demonstration.